So hello everyone and welcome to Science in the Bible speaker series. We're very happy to have Dr. Michael Behe here to speak to us, and it's a blessing to have him here, so thank you for joining us. Some brief club announcements. They're not many. We're going to have a speaker in November. We're not sure of the date and time. We'll send that out. Dr. Tinglu Yang's speech that he gave 3 weeks ago now is up on YouTube. So if you want the link, I'll send it out in the next email. And it'll be generally available. Just, look up Penn State Scible, Science in the Bible Club on YouTube, and we'll be there. So now I'd like to start off with a prayer. In the name of the Father, and Son, and Holy Spirit, amen. Thank you, Jesus, for allowing us to gather in fellowship safely during the pandemic and allowing us to host speakers so that we can discuss the science of the natural world. Please grant us clarity of mind and the drive to learn and understand the world more fully. In Jesus' name we pray, amen. So briefly, I just want to tell everyone here about the format of the Q&A. So the way that it's going to work is that there's a Q&A function on the bottom of the Zoom. If you want to ask a question, you can pose it there. If you would like to join, if you'd like to ask a question, put it in there. And during the last about half hour of the talk, I'll ask the question to Dr. Behe just to eliminate, questions that are redundant and to promote more challenging and interesting questions. Now, if you feel that I didn't do a good job asking your question, then just message me and then I can let you speak as well. Hello, Dr. Tinglu Yang. It's nice to see your whole group joining us. So thank you, Dr. Behe. I'll hand it over to you now.
Okay, thanks very much. Let me just talk for a minute about myself, and then we'll switch over to the science. I'm going to be talking about evolution, and most people think that most people have objections to evolution based on religious considerations, and that's certainly true for some folks, but it's not true for me. I'm a Roman Catholic, and I was born into a Catholic family, and I went to parochial schools through high school, and I was taught Darwinian evolution in high school, and we were taught kind of a theistic evolution-y type of Darwinian theory. That is that, God could make life however he wanted to, and who were we to tell him otherwise? And it seems from what we can see that God used natural laws, the laws he set up in nature, to make life. And that always seemed fine with me. So I actually thought Darwinian evolution was true, not because I knew anything special that other folks didn't, but just because my teachers told me it was true. You're supposed to be learning from them. And it was not until the mid-1980s, which probably strikes you as a long time ago, some of you folks, that I read a book called Evolution, the Theory in Crisis by a man named Michael Denton, who was a geneticist and medical doctor in Australia. And he pointed out a lot of problems for Darwinism that I had never thought of. And it startled me. And from then on, instead of just accepting Darwinism, I looked for evidence and looked for papers published in the scientific literature that would support its claims. And what I found is that, when you study biochemistry, you study fantastically complex cellular systems. And I had frequently, before then, asked myself, gee whiz, I wonder how that thing could have evolved. But before reading Denton's book, I followed that statement saying, well, I guess somebody knows. And the assumption that somebody knows kind of defeated or stopped me from thinking further. But after reading that book, I asked, well, who has shown how these systems could evolve? And a quick trip to the library showed that nobody had. It was It's all, it was all assumption, all in people's minds. And so from then on, I became very interested in evolution, and in particular in intelligent design, because when you come to doubt Darwinian evolution, intelligent design, pops into your head pretty quickly. So I just wanted to say I'm offering these not so much as a Christian or as a religious person, although I am, and although these results, I guess, do have some bearing on theological ideas. But I'm a biochemist, and so I'm offering them mostly from a scientific, biochemical point of view. Okay, so with that, I'm going to start to share my screen and put up my PowerPoint. Okay, let's see here. I'll do that. I think that'll do it. No, maybe not. There we go. Okay, let me just arrange things a bit. Okay, and let's see. Can I get sorry. I'm just looking around for a pointer. I know there's a pointer here somewhere, but let me see. Hide, optimize. Recording cloud. Okay, well, I can't see it, so I'll just use the cursor to point to places that I want to draw your attention. So the title, as you can see, is The Biochemical Argument for Intelligent Design. And that's exactly it. It's going to be that, in my view, what science has discovered at the molecular basis of life strongly indicates purposeful intelligent design. And I have, for the past nearly 25 years or so, I've written 3 books. First, Darwin's Black Box in the 1990s, The Edge of Evolution in 2007, and most recently, Darwin Devolves. And they make 2 basic points. The first point is shown on the screen here, that Darwin's mechanism is grossly inadequate to explain the molecular structures of life. Since science has discovered the intricacy and elegance of life, it's become clear to those with eyes to see that Darwin's simple mechanism of random changes and selection will not cut the mustard. The second, that point is necessary to make strongly and to make first, because most people assume that Darwinism has been shown to be true, just like I assumed it before I read that book in the 1980s. But once you have gone beyond that, the second point is that a firm conclusion of purposeful design is empirically justified. That is, design is not some default conclusion after you've done away with Darwinism. Rather, intelligent design, purposeful design is clearly apparent from the physical aspects of what we've discovered. So let's take a look here. We will start with the subject matter of the first book, 1996, Darwin's Black Box. And its starting point is an idea that Charles Darwin himself wrote about in The Origin of Species. On page 158, in a section called Organs of Extreme Perfection and Complication, Darwin was considering the eye, which even in those days, in the mid-1800s, was known to be an extremely complex and elegant system. And he went through a brief discussion pointing to different kinds of eyes in the animal kingdom and said, well, maybe they might have been intermediates for each other. It was not really a detailed analysis. But he said, well, hey, it doesn't seem to him to be such a big problem. And he ended that section on organs of extreme perfection by saying this, that if it could be demonstrated that any complex organ existed, which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down, adding, but I can find out no such case. So here he was insisting that his theory had to work very slowly, step by step, in a gradual manner over very long periods of time, because he knew that if there were sudden leaps in life, there were sudden increases in complication or increases in something, then it would look suspiciously as if something other than random changes were involved. So Darwin always insisted that life had to be built up by a gradual manner in numerous successive slight modifications. Well, okay, let's take Darwin at his word and say, well, what sort of a system? We won't say could not possibly. That's that's setting up an impossible standard. Nothing gets proved in science, but we can ask ourselves, what sort of a system sure doesn't look like it could be put together by numerous successive slight modifications? And one that fits that bill is one that I called irreducibly complex, or something that had the property of irreducible complexity. An example of that from our everyday life is all around us. For example, you consider a mousetrap, just a simple little mousetrap. It's got a number of parts. It's got this wooden platform here, and it's got this spring which has an end which hooks around another metal piece, and that's stabilized by this other metal piece called a folding bar, and here's the catch. And it turns out that all of these pieces are needed. If you take away, say, the hammer, you take away the spring or the catch or any of these guys, then you don't have a system that works half as well as it used to, or it doesn't work a quarter as well. It's broken. You have a broken system. Turns out that this mousetrap needs all these pieces to work. So if you wanted to evolve something like this mousetrap by something like a Darwinian process, how would you do it? Natural selection needs something to select, something that helps, something that improves. And if you're starting with, say, just the platform, that's not going to catch mice. And if you add the holding bar, that doesn't help. You have to have the whole thing pretty much together before you get the function. And so that's a huge problem for Darwin's theory. Anytime you need to take more than one step to get an improvement, if you need to take a step but that doesn't help or actually hurts, and then another step, and that would help, then that's a big problem. Any unselected step is a big problem for Darwin. Okay, now in the past 50 years and more, science has discovered a lot of systems in the cell that in fact are irreducibly complex. And that's because science has discovered that the cell is run by molecular machines. Mousetraps are machines, and they're very, very simple machines. And the machinery of the cell is much more complex and elegant. And here's, I think, one of the best visual examples of a molecular machine. It's called the bacterial flagellum. which is quite literally an outboard motor that bacteria can use to swim. It's got multiple parts. It's got this long filamentous structure which acts as the propeller. And it spins around and around and around, and it pushes against water, which pushes the bacterium forward. And the propeller is attached to the drive shaft, this yellow thing, by a hook region, which it acts as a universal joint, which allows the rotation around the drive shaft axis. So this spins around here like this, but magically, when this comes up, this spins in this direction. So it's not, if this turned around, you might think that this whole thing would have to whip around like that. But no, with this joint, it transfers the rotation from this plane into that plane. The drive shaft is hooked up to the motor, and here's a number of proteins that act as a stator, which are like clamps that hold the whole apparatus in place as it spins. There are other proteins which act as bushing material, allowing the drive shaft to poke up through the membrane, and many other things as well. And let me tell you that this is a cartoon version of the flagellum, that these parts are not little geometrical shapes. They're all themselves very complex proteins. Nonetheless, I think you can see that like the mousetrap, the flagellum needs a lot of parts to work. If it weren't for the propeller, if it weren't for the drive shaft or the motor or the hook or any of these parts, the thing wouldn't, it's not like it would spin half as fast as it used to. It wouldn't spin at all. It'd be broken. And so like the mousetrap, it's very difficult to see how this could be put together by numerous successive slight modifications that Darwin always insisted on. So the point is that irreducible complexity is a huge problem for Darwin's theory, and many, many, many things in the cell are in fact irreducibly complex. Okay, let me see where I'm going. Okay, so it's a problem for Darwin's theory, but what does that have to do with intelligent design? Well, back in the 1700s, this guy here, a guy named Thomas Reid, he was part of what was called the Scottish School of Philosophy, and he wrote a lot about philosophy of mind. And he wrote about something that he called the design principle. And he said that, you know, you can't see intelligence. You don't look at a person and see, you know, lights coming on and off in their heads. Rather, he said, the general principle, the design principle, is that intelligence in the cause can be inferred from its marks or signs in the effect. We know that something is intelligent if we see it doing intelligent things. And we know how intelligent it is if it can do things that require more and more intelligence. Well, okay, what does that mean? Let me take a peek ahead. So, okay, so that's the design principle. And he says that, you know, we infer intelligence by the marks of intelligence. Well, what are those marks of intelligence? What are marks of design? If you look in a dictionary, design has a number of definitions, but the pertinent one for our purposes is this, that design is the purposeful or inventive arrangements of parts or details. And we can simplify that and say that design is the purposeful arrangement of parts. And that means that we infer design, we suspect design, or we decide that design has happened whenever parts appear to have been arranged for a purpose. So a purposeful arrangement of parts is how we detect design. And the strength, there are two other points that are necessary to keep in mind, or you can be misled. The first of these is that the strength of the design inference is quantitative. That means the more parts that you have and the more closely they are arranged for a purpose, the more and more and more confident you can be that it was purposely designed. The second point is that design can be found at multiple independent levels. So some particular piece could be designed and it could be put into a larger assembly where the assembly is designed, and then assembly can be used to in the design of a higher system. So example, a bolt could be designed, and the bolt could be put into to hold two pieces together into a machine, and the machine itself could be part of a factory, and so on. So design can be found at multiple independent levels. Okay, well, this is a bunch of words. Let's look at an example. Okay, let's put this to a test here. Now, this is a Far Side cartoon. And in the cartoon, we've got a troop of jungle explorers, and the lead explorer here has been strung up and skewered. And this fellow with the glasses here turns to this guy here and says, "That's why I never walk in front." Words to live by. Now everybody here, everybody viewing this, immediately realizes that this was designed. This was not an accident. It was intended. How do you know that? Well, you know that because you see a number of pieces arranged for a purpose, a grisly purpose, but a purpose nonetheless. So what we see is the purposeful arrangement of parts. Okay, so that's how you recognize intelligent design. As a matter of fact, a purposeful arrangement of parts is the way, the only way that we recognize the work of a mind. You have to understand that with design, there is an additional component besides matter and energy and laws of nature and so on. There is purpose. There is intelligence. That's what design is. It's the workings of a mind. And since we ourselves are intelligent, we can recognize purpose. We can recognize when some things look like they were arranged to do something for a purpose. And so that is the way that you recognize a design. And now we can say that irreducible complexity is a twofer. That is, it's got two important features that are relevant to our purposes here. The first is that it is an obstacle to Darwinian evolution. You can't put together something like a mousetrap gradually or a flagellum gradually. And the second thing is that it is evidence of purposeful design. If you need multiple pieces for a purpose, then you can recognize the purpose in the arrangement of parts. So irreducible complexity is both a problem for Darwinism and a very strong pointer to intelligent design. Okay, so that was my the gist of the first book that I published on this topic back in 1996. I'm going to skip over the points that I made in the middle book, The Edge of Evolution, and go right to the newest stuff that I wrote about in 2019 in Darwin Devolves. And let's see here. The most important thing in Darwin Devolves is to realize that science advances with new technology. Our senses are, pretty good, but not good enough for to investigate all of nature. An example is the invention of the telescope. If before the telescope was invented, astronomers were, not very good at figuring out how the universe worked. But after the invention of the telescope, the universe was seen to be much larger than was known, and the bodies in space were seen to move in different ways, and lots of relationships could be discerned that could not be discerned before. The big breakthrough for biology was the invention of the microscope in the 1600s. The microscope allowed things to be seen that simply couldn't be seen before. Before the microscope, insects were thought to be featureless entities that didn't have any internal organs. And, as far as they knew, that had to be true. You couldn't see them. There was nothing to see. But with the invention of the microscope, you could see all sorts of things like compound eyes of insects and many, many details that showed that even these tiny creatures were very complex beyond our imagination. And so science always advances rapidly when new technology is invented to investigate an area that couldn't be investigated before. Turns out we're living in an age like that. And in particular, in the age of something called the DNA sequencing revolution. Now, we didn't even know that DNA was the genetic material until the late 1940s. Remember that Darwin himself didn't know how heredity worked, even though his theory depended strongly on the details of that. So Darwin was actually working very much in the dark. And it wasn't, as I said, until the late 40s that DNA was discovered to be the genetic material. And of course, Watson and Crick then discerned the shape of DNA, and the genetic code was discovered. That is where the sequence of bases, nucleotides along the double helix is a code that determines the sequence of proteins, the sequence of amino acids in proteins, which then tells the protein how to fold and assume the shape of the molecular machine that it's destined to be a part of. So all the information, say for the bacterial flagellum, the components of it, the proteins that make it up, reside in the sequence of nucleotides along its DNA. Well, for the longest time, people couldn't decipher the sequence of DNA, but over decades, progress was made in being able to do that. And here's a typical sequencing run where each band of these tells people what the next letter is in the DNA, and here's a readout of all these letters of a piece of DNA. it turns out that just like computers, which in the 1940s and 50s were big and clunky and expensive, so were the methods to sequence DNA. And it wasn't until in the 70s they were still clunky. In the 90s and 2000, they were struggled, and they just sequenced the human genome. Now people spit out sequences in a week or two that would have taken decades or would have been impossible to do not too long ago. Not only human sequences, but this entire genetic complement, the genome of all sorts of organisms: people, dogs, kitty cats, chimps, mice, horses, birds, salamanders, bacteria, viruses, everything. And you can compare them to each other. And now you can ask questions about the sequence of DNA that helps you to decide how much Darwin's theory can do. The interesting thing is that what evolution is doing, or what is happening, in the important changes that are supposedly fueling evolution are molecular changes in the sequence of DNA. What happens is that a mutation is a change in one of these nucleotides because of an accident when a piece of DNA is being replicated, when a cell is duplicating. Maybe one of these changes, or one is left out, or a whole chunk is left out, and this is joined to here. But the point is that mutations are molecular changes. They are molecular events. So in order to understand Darwin's theory, you have to look at the molecular level of life. You can't just look to see if brown bunny rabbit parents give birth to a white bunny rabbit offspring and say, look, there's a mutation. We know that Darwin's theory works. Rather, you have to look at the molecular changes. That's where the real action is taking place. Okay, so in order to understand, for example, if a car works, you just can't look at the doors and the surface and see how pretty it looks. You gotta open the hood and look at the nuts and bolts in the engine. Well, this DNA is the nuts and bolts in the engine of life. And so using the great amount of information that science has come up with in the past 20 years, just since the year 2000 or so, Darwin makes three key points. And I think I'll probably only have enough time to talk about the first one, called the first rule of adaptive evolution. But I hope to be able to get to some of the other two as well. But I think this is the most important one, the first rule of adaptive evolution. That asks the questions, well, what is the first thing that's likely to happen if an organism has to adapt to a new environment or a new set of circumstances? And much of the work through which one can answer this question has been done by this guy. He's a man named Richard Lenski, who's at Michigan State University. And 30 years ago, he started an ongoing experiment where he grew flasks of E. coli, the bacterium E. coli, in his lab in Michigan State. And he started with 12 flasks that he kept for replicability's sake, and he let the 12 bacteria in the 12 flasks grow overnight. And it turns out that E. coli is so small and reproduces so quickly that overnight it will go through 6 or 7 generations. And in his small flasks, there were 100 million or so bacteria. And so he let them go overnight. And so they'd go through 7 generations, produce a large number of bacteria. And he'd come in the next morning and take a little bit from each of the flasks and put them in another 12 flasks and let them go overnight, and come in the next morning and let them go overnight, and do it every day for the past 30 years. And it turns out that in that time, the bacteria have gone through 60,000 generations, and there have been trillions of bacteria born and died in his flasks. And he did this just to see what would happen, to see how they would evolve, how they would change in his flask, in his labs. And in the beginning, back in the 1990s, all he could do really was to look at changes in the shape or the behavior or some observable property of the bacteria. So some bacteria got bigger and they grew better. So he was able to write a paper saying, well, these bacteria evolved and they're dominating, and this is what they look like now. But he couldn't tell the molecular change that was happening, simply because the technology for sequencing DNA was not available back then. And this occurred for 10 years or so, that he would look at properties of the bacteria, but he couldn't tell the mutations that were occurring. But then around the year 2000, he was able to track one mutation down. He saw that some bacteria were growing much, much faster, much better, and taking over the flasks in his experiment. And he was able to track down where in the genome, what pieces of DNA were changed. And it turns out that the mutant bacteria had lost the ability to metabolize a sugar called ribose, which was really strange. And the reason is that there was a deletion of a large chunk of DNA that contained the genes to allow the E. coli to metabolize ribose. And with that missing, for some reason, the bacteria grew much better. So here he says the expression of both the ribose operon and the maltose regulon, that's another sugar, decreased after 20,000 generations. Therefore, these reflect beneficial mutations. However, they were deletions of the ribose operon. So here's something that, seemed a little unintuitive, that throwing something away, losing something, helped the bacteria survive in those flasks. Later on, as sequencing became easier, he was able to track down a bunch of other genes whose where mutations in them also helped the E. coli to grow better. And here, these are 4 different genes. These long bars here are supposed to be the DNA sequence of the gene. And you can see little arrows pointing down to this one. And these arrows represent places in this gene in at least one of the 12 replicates that he was growing where a mutation occurred. So he saw that in one replicate, maybe a mutation occurred here and nowhere else. But in a replicate, another of the replicates, maybe it occurred here or maybe here. And it turns out that this, if you modify, if you mutate any of many different sites in a gene and get an improvement, that's the signature of a mutation that is working by either degrading or destroying the activity of the protein. Because you can break a gene at a lot of different places in its length. But if you were going to try to improve it, there would probably be only one or two places where you could do that. It's kind of like taking a hammer and hitting your computer. There are a lot of places where you can hit your computer and break it. Not too many where you can hit it and improve it. So if you can mutate at a lot of different places, then that means that you're very likely degrading or destroying the gene. And he saw that each of the 4 genes that he looked at had that pattern, that they were had multiple changes that were effective. Well, I reviewed Lenski's work along with the work of a lot of other experimental evolutionary biologists, and wrote up a review paper in something called the Quarterly Review of Biology back in 2010. And the paper was titled Experimental Evolution, Loss of Function Mutations, like those genes that we just talked about, and, quote, the first rule of adaptive evolution, close quote. And from those results, I proposed we could think of those as kind of a rule, a new rule. And the first rule of adaptive evolution is this: break or blunt any functional coded element, and by that I mean a gene or a control region or something, any functional coded element whose loss would yield a net fitness gain. Evolutionary biologists call the offspring fitness for some reason. So a net fitness gain is essentially just, grow faster and you leave more cells, more organisms. And so the first thing the first thing that's very likely to happen if an organism is adapting to a new environment is to break or blunt any gene that would help. Okay, so if, like with the ribose, it helps to get rid of a gene in some circumstance or other, then out it goes, and it goes out first because it's a lot easier to break a gene than to improve a gene. Here's a little slide trying to illustrate that point. Here's points where a gene might be broken. Most genes are, say, 1,000 nucleotides or more long, and any place, you change them in many, many, many places in that length, you break it. But if you wanted to improve a working gene already, there's almost always just one or two or very few locations where if you changed it, it would help. So the first rule of adaptive evolution says, because it's so quick, these helpful mutations will arrive a lot faster than these. So the first thing you'll do is get rid of any genes that are slowing you down in your new environment. So, so that's one of the main message of Darwin Evolved. That is, it's a lot quicker and easier to break a gene than to build one. And because breaking genes actually is adaptive oftentimes, then that will dominate evolution. Some people say, well, how can throwing something out, how can getting rid of something be helpful? How can you have helpful degradation? Well, suppose you own this really pretty spiffy car and you were driving, but at some point it turned out that your life depended on your nice car getting a little bit better gas mileage just a bit better gas mileage. And if it didn't, you would die. Okay, so your life depends on this. What could you do quickly to help your car get better gas mileage? Well, you can throw out the doors, you can take off the hood, you can remove the hood of the trunk and the windows and the seats and so on. Of course, this doesn't look anywhere near as pretty as this, and doors and so on are useful in many circumstances. But if your life depended on getting a little bit better gas mileage, this is the way to go. Everybody who hung on to their pretty car is now dead, and you are living in the road warrior world, post-apocalyptic. So degradation can be helpful. This car has adapted to a new environment where fuel economy is the highest priority. So losing a bunch of different things is actually helpful. Well, in the year 2016, sequencing was in its full flower. And Richard Lenski and co-workers were able to sequence the entire genome of many different E. coli from his experiment and look at the top 30 genes that were most strongly selected, which means they helped the bacteria the most. And they were able to look at the mutations in the genes. And here is a table, table 1, that had the highest selected mutated genes. And here are the number of mutations that occurred in them. And you see that in all of these guys, there are multiple mutations that are helpful. Nonetheless, as we said, this is the signature of mutations that help by degrading a gene, not by improving the construction of it or by giving it new features or anything. So all of these are likely to be degradative mutations. There is a second table in his paper, and these are mutations, larger mutations, where big chunks of DNA are deleted or inserted where they're not supposed to be, or other such things. And they were very helpful as well. So essentially, the 30 best mutations, the ones, well, the mutations that helped, all of them were degradative. All of them decreased the genetic information found in DNA. So this is what observation, not theory, this is what observation demonstrates about random mutations. We already knew this point, that of those mutations that affect an organism, those that are either helpful or hurtful but not neutral, about 99% are detrimental. That is, they hurt, they actually hurt. But of even that tiny fraction that helps, of even beneficial mutations, the great majority break genes or degrade function. This is new. This is what we didn't know before, that beneficial mutations can arise by breaking genes, and they're a lot faster and easier to come by. What's more, this process is, as I have here written, as relentless as the tide. This principle, the first rule of adaptive evolution, the rule is not a law, it's not an ironclad law, it's like a rule of thumb. This is what you should bet on, this is what you should, will happen most frequently. This is not to say that something that looks constructive is absolutely forbidden, but that it's overwhelmed by a rain of helpful but degradative mutations. Just like, you can build something, but then the tide will come in and wash it away. And an illustration of this is that there was one mutation in a gene for helping to transport a chemical called citrate, and I don't have time to go into it, but it looked to some of Richard Lenski's group as if it might be constructive, and a big deal was made out of this. I don't think it was at best marginal, but let's leave that behind, because after that new mutation came in, other mutations occurred to support that first change in metabolism. And those changes, those new mutations that came along, were the loss of another gene and the loss of this gene, ACE-A. And other genes were lost as well. So, the point of the first rule is not that, everything has to be degradative, but that, you have degradative mutations first, and if something that's not really degradative comes along and helps, well, that's fine. But right after that, these degradative mutations can swarm to help the organism adapt to its new metabolism. Okay, let's see. So Darwin's mechanism is dominated by what I call poison pill mutations. That is, positively selected, helpful, beneficial mutations that destroy genes, that cause them, the genes, to lose their function. And it works, Darwin's mechanism works by squandering genetic information for short-term gain. Now, this can be important. This actually can help organisms adapt to new environments, new circumstances, but it's not something that's going to build sophisticated machinery in the first place. Okay, now I'm going to, let me just put this up. And it's not just bacteria. This degradative process, Darwin's mechanism, is a degradative process. And everywhere we look, everywhere I have looked, the changes, even the ones most highly touted by Darwinian biologists, turn out to be degradative changes. Now that we are able to sequence DNA and look at the molecular changes that cause them, it turns out that they're degradative ones. For example, puppy dogs. Dog breeding has been pointed to by evolutionists for a long time as showing the plasticity and changes that can occur by selection. And that's true, but I don't have time to look at it. The genes which affect those changes have been tracked down because some biologists are interested in dog breeds as examples of evolution, and the mutations associated with those changes have been discovered too, and the great majority are degradative changes. The polar bear. The polar bear is thought to be descended from the grizzly bear, the brown bear. It turns out that the entire genome of the grizzly bear and the polar bear have been sequenced and compared to each other to see what genes had to change in order to get yourself a polar bear. And here's the paper in which it was first done, and the authors write that a large proportion of mutations were predicted to be functionally damaging, predicted by a computer algorithm. And it's actually more than 50%, for reasons I describe in my book. So again, yeah, maybe a polar bear did descend from a grizzly bear, but it was devolution, not evolution. It occurred because it was losing genetic information, not gaining genetic information. And I guess this is what I'll leave you with, and I'll stop after this slide. This is Darwinian evolution at work. This is an illustration. There's a picture I got off the internet, and it shows some soldiers in a South American country, and here's some army vehicles coming up. Turns out that up this road, there was a drug growing operation, and the army was informed about it, and they sent some troops to shut it down. But the drug growers got wind of the operation, and they blew up the bridge of the only road that led to their outfit. Now, from the point of view of the drug growers, this is a beneficial change. This is a beneficial mutation because the army can't invade them now. The bridge through which they would get to them has been blown up. So the drug growers can run away and live to do more bad stuff another day. It turns out that Darwinian evolution, Darwinian processes, do this all the time. That is, they blow things up which are either hurting in the current circumstances or might help if they were blown up in the current circumstances. But again, blowing up a bridge does not tell you how a bridge is built. And this is not the inverse process of building a bridge. So Darwinian evolution is important. It does have medical consequences and other things, but it is not a process that can explain how this sophisticated, elegant machinery of life came about. Okay, let me go down to the bottom here and just say that there, let me put that up. Now, as you might have known, I've had a few critics of my books, and not everybody agrees with this. And they've written a lot of criticisms, and I've responded to. And my responses can be found at my university website. These are mostly scholarly papers. They can also be found at the website of the Discovery Institute. I have a lot of stuff there. And also, I've got a website myself, michaelbehe.com. A lot of responses, rebuttals, and so on. So if you really want to delve into this and see, you know, Behe says this, what have other people said, and what has he responded to this, and so on, that's the place to go. Also, in November, in a month or two, Discovery Institute is going to publish a collected volume of my responses to critics. So that'll, if you're interested in more details, that's the place to go. So I think I will stop my sharing and go back, and I'd be glad to address questions.
Yep, so just thank you very much to Dr. Behe. I think I can give you a round of applause. I'm not sure if any of the participants can. But for questions and answers, if you have a question, please put it into the question and answer box, and I can ask it for you. And if you feel as though I'm not asking your question well, just write me a personal message over the Zoom, and then I can give you the ability to speak. There are some questions already, so I'll ask those, and if you have more questions, please put them in the Q&A. So I think this one, which I think is very pertinent because of the current crisis going on, it says, so you mostly talked about point mutations and different deletions. However, you didn't talk about insertions like viral insertions, gene duplications, or transposons. given that the coronavirus that we're currently seeing is a result of or the data indicates that it's a result of a recombinant chimera of two different viruses, what do you think how does that interact with your theory?
Well, as I said, not all mutations are destructive, but you've got to be real careful when you're counting constructive mutations. Let's take gene duplication. Some people say gene duplication is a way to get another gene, and that's constructive. And many scientists think that that's a route to making new genetic information. But copying a gene just gives you another copy of itself so that you have two identical pieces of DNA. And that does not explain where the original copy came from. And even after you have two, you don't have anything new. It's just another copy. Sure, you might be able to, in theory, change this and let this keep its own activity. But the question is, can you do that through a random process, and how far can you go? And in my book, Darwin Devolves, I have a section talking about the severe difficulties, even if you've got a duplicate gene, of now modifying it into something different. Sure, you might be able to get one, say, taste receptor and duplicate it and get a second copy of it, and they pretty much do the same thing, but the first one did it beforehand. But you won't be able to get the different genes for, say, the bacterial flagellum for that, or different genes for the ribosome for that, or other such things. As far as the COVID-19, the coronavirus goes, it might be a hybrid of two different viruses. All that is still speculative and uncertain. But even so, even if it were, it's still got the basic machinery that it always had. The hybridization might affect the ability of one protein to bind to a human receptor protein, but that's a little change. It's not a change in the function of the protein itself. And if you look in the literature, you find that coronaviruses are not new entities. They did not arise just in the past few decades. Some people estimate them to be several 100 million years old. That is, the same structure of the virus, same basic structure, is several 100 million years old. And that's the case with most viruses. And the origins of viruses themselves is completely up in the air. There are some speculations, but nobody really knows. So there are smallish mutations that do affect human health, but they are not the kind that can explain complex molecular systems.
So thank you for that. Another question is regarding the Lenski experiments with E. coli. If the E. coli undergo destructive evolution, could altering the environment of the E. coli that it is grown in could that favor constructive evolution? In vitro experimentation can't always represent what's happening with external factors. For example, electromagnetic waves have been shown to alter mutations.
I'm going to ask you to repeat that because I didn't quite catch it.
Okay. So if altering the environment that the E. coli is growing in, could that favor constructive evolution? And then there's a comment that says, in vitro experimentation can't always represent what happens with external factors. For example, electromagnetic waves have been shown to alter mutations.
Okay, well, can you just say that first part again? Not the comment, but the
Okay.
question.
In the Richard Lenski experiments, the E. coli undergoes destructive evolution. Could altering the environment favor constructive evolution?
Okay, thank you very much. And the answer is no. Altering the environment does not favor constructive evolution. Whatever change you make, whatever change you make, the fastest mutations to come along, just because of statistics, the fastest ones are degradatory, just because there are so many ways to degrade a gene. And if one of them helped in some way, then it would automatically be selected by natural selection. Natural selection, as we are always told, is blind. You know, it's a blind watchmaker. So it doesn't care whether a helpful effect is due to a broken gene, a duplicated gene, a new gene, whatever. So you can't stop it from being selected. And I did have that one slide or a couple slides during the talk, the relentless as the tide slide in the next couple. And that is that when one change, the environment changed, you can think of the situation as the environment of the E. coli changed because a mutation came along that allowed it to metabolize citrate, which the ancestor bacteria could not. But there was this change, a mutation that I said got a lot of attention that allowed it to eat citrate. Now it had a lot more food than it did before. So hey, is it going to start constructing genes to help it with that? And as I showed, no. The immediate effect was to select, a half dozen more degradative mutations, because when it was metabolizing citrate now, it would help to get rid of this gene and to get rid of that gene and the other one too. So there are no circumstances that favor constructive changes.
Okay, so the next question is, we hear a lot about bacteria that have developed resistance to most antibiotics. They are sometimes described as superbugs. We are given the impression that humankind is in peril, but based on Darwin devolves, is it more likely that these are damaged bugs and unlikely to pose a long-term threat?
It depends. Some of the bacteria resistance mutations are actually transmitted to the pathogenic bacteria from other bacteria in the wild. That is, bacteria have a capacity to, say, give pieces of DNA in the form of things called plasmids, small circular pieces of DNA, from one to another. And it turns out there seem to be antibiotic genes floating around already in the biosphere. And so some very resistant bacteria have picked up genes from the environment. And that is not degradative mutation, but that doesn't explain where the genes came from in the first place. It doesn't explain how they got into the biosphere. Somebody's just giving them a copy of it. There are other resistance mutations that occur through degradative processes. For example, some antibiotics work by sticking to a particular molecular machine and messing up its function in a pathogenic bacteria. So if the protein of the molecular machinery in the bacterium is mutated, sometimes it kind of has a little peg sticking out of it, you can think of it as. And so when when the antibiotic goes to bind to it, it's not able to bind to it because the shape of the surface of the bacterial protein has changed. That's degradation. A bit, well, it's not degradation, but it's not certainly not construction. But that is not that does not explain construction either.
Thank you. Another
Sure.
question is, does genetically modified food cause trouble to the human body?
Well, not to me. No, I eat it all the time. Uh, um, there's a big of course there's a lot of controversy on that, and I don't see I see no reason why it would cause any hurt to the human body. One thing that should make people careful is that, or try, more hesitant to make genetically altered crops is, suppose the gene gets into the wild or the species spreads beyond what it was intended to do. What if the modification gives it an advantage somehow and it displaces native crops or displaces native plants and so on? Biology is a very complex system, and sometimes you don't know what effects something is going to have, and so you have to worry about unintended consequences of these things. But for the most part, eating them is not the problem, at least in my view. It's the uncontrolled spreading of them getting out where they weren't intended to.
Thank you. Now, this one is a little bit more in depth. So, and based on arguments is what I mean by in depth. So, Francis Collins, who's the current director of the National Institute of Health, and he's a religious scientist as well, and he says that the intelligent design argument is sort of a God in the gaps argument, meaning that the gaps that science can fill in evolution are filled by God. And he says this is a dangerous idea that he lends caution to, because if those gaps are filled by science, then religion as a wholesale is discredited. So what are your comments on that?
Well, I disagree. And can I share my screen again here?
Absolutely.
For this answer. Here, let me zip on over to my slides here and just bring this guy up here. Oops, sorry. There we go. Okay, so you look at that and you say this trap was designed. Is it a God of the gaps argument? Is concluding that this trap a God of the gaps argument? Because after all, there might be plants that grow like that, and the answer is no. This was designed not because of a lack of evidence, but because you see the arrangement of the parts. Intelligent design is not a God of the gaps argument. It is a positive argument based on empirical data and logic, the same logic we use every day in our lives to see that things were the result of intelligent activity, the result of a mind. I admire, let me unshare this here. And I admire Dr. Collins. He's a brilliant guy, and he's a very strong Christian. And I watched his Templeton Prize address just the last week or so. He's very eloquent. Nonetheless, he has no answers to the questions I raise. And he, in his book, The Language of God, I think it was the mid-2000s somewhere, he did complain about the intelligent design movement, as you say, but he famously offered absolutely no ideas about how Darwinian processes could build the machinery of life. So two things, intelligent design is a positive argument based on empirical data. It is, in that way, it's a scientific argument. And it's not what's called a god of the gaps. And in my view, it's simply name calling to try to defend the status quo.
Thank you. This is a question from me. So you talk mostly about protein coding. However, a large portion of the way the cell machinery functions has to do with non-coding regions, promoters, and transcriptional factors and whatnot. So does that fact interplay with the intelligent design argument?
Well, that's a good point. Most of the genome is not protein coding, but consists of regulatory regions and programs to time the expression of genes and so on. I think that fits right into the devolutionary aspects of Darwin's mechanism. You can change an organism. Any way to change an organism will be selected if it arrives most quickly and if it's beneficial. So suppose you had some sort of a promoter that turned on a gene, and it'd be better for that gene not to appear. Well, you can, as Lenski's work showed, you can mutate the gene and mess it up, or you could get a mutation in a promoter and destroy the promoter so that the gene isn't expressed. Or you could destroy a repressor and have the gene expressed all the time, if that would be beneficial. Anything that changes, anything that affects the survival of the organism in an environment will be selected. And anything that you can think of in biology, if it's broken, even if it's not just a protein coding gene, if it's a enhancer or a modification site or anything, and if it's destroyed, if that were helped, it's fair game for being selected. The basic problem is that all of these things, not just protein coding genes regions themselves, but the promoters and so on, consist of a fairly complex module of nucleotides. There's information there, and it's a whole lot easier to destroy the information than to make it. So they'll be fair game for destruction too. Additionally, you can think of that another way and ask yourself what changes in promoters and so on in Richard Lenski's experiment were selected. Not many. There might have been one, but not many. Additionally, in my book, The Edge of Evolution, I looked at the changes in malaria and chloroquine resistance and malaria's development of chloroquine resistance. And in an astronomical number of possibilities, no changes in regulatory regions, no changes in the non-coding DNA were helpful. There are only 2 crummy point mutations in a particular protein in malaria helped it resist chloroquine. So it's easy to say, well, we don't know much about that. Maybe that's where things are going on. But now we have evidence because of our ability to sequence DNA and all these great studies, and we don't see any trace of those more exotic effects.
Okay, thank you. And so you would, just a brief follow-up, you would expect that promoters, if they were to change, they would be destroyed, or their function, they wouldn't bind as well, they wouldn't be as effective targets.
I'd say that is, most favored statistically. You might by accident just mutate a sequence, and voila, you've got a new promoter there. And it just happens to turn on this gene, and that's helpful. Okay, great. But the next change that comes along has as great of still has a better chance of being degradative than constructive, and the one after that, and the one after that. If you're building a house and each step you take, you have a 10 times greater likelihood of breaking something than putting a new piece of lumber in place, your house isn't going to get built.
Okay, thank you very much. Another question is, with miracle healings, there are numerous cases of tumor disappearing instantly. What is the best theory of what is happening on the molecular level? And do you think that the constructive mutation is caused by God would occur, or is something more complex going on?
The short answer is I don't know. I don't know much about miraculous healings or the stuff, let alone the molecular events underneath. God has his own ways, and he can do whatever he would like. But I just work with the results that are common to the scientific community, and there's plenty enough there to see the purposeful design of life, and other things like mentioned in the question. We might be able to get an answer in time, but we don't have any good data right now.
Thank you. One more question, which I think will be our final question, is in your book, you talked about how you calculated the computationally the time it would take to form disulfide bonds and sort of minimum irreducible complexity. At that time, were you a proponent of an intelligent design theory, or did you see this data and say this must be intelligent design?
At the time, I was an intelligent design proponent. That was, at least 10 years. When David Snook and myself published a paper on that topic, was 10 years after I had written Darwin's Black Box. So when you no longer take Darwinism as a given, and you are asking questions of whether it can do things, it opens up a whole lot of questions. So that one, my strategy or my idea is to look at the easiest things that might be done with Darwin's mechanism. And it turns out that it can break things or change single things pretty easily. But as soon as you need two things to happen at once, like the disulfide bond or chloroquine resistance in malaria and so on, then it immediately becomes exponentially much more difficult and starts quickly to break down.
Okay, thank you very much. I think we're just about out of time. I just want to take this time to thank Dr. Behe for joining us and giving us a wonderful seminar.
Thank you.
Yeah,
okay,
so yeah, just a reminder, this has been recorded, and I'll be sending out that link and editing it accordingly and posting it to YouTube, so that will be available. And the previous one is available as well, and there will be a speaker in November. So again, thank you very much, and everyone enjoy the rest of your day.
Bye-bye.